There are moments in science that divide history into a before and an after. The discovery of fire, the telescope, and now, perhaps, a mudstone rock sitting in a sealed titanium tube on the surface of Mars. You've likely seen the headlines. NASA's Perseverance rover may have found the first evidence of ancient life beyond Earth, not on some distant exoplanet we'll never visit, but on our immediate neighbor, just 140 million miles away at its closest point. If confirmed, we're not talking about the greatest scientific discovery of the decade. We're talking about the greatest discovery in the history of our species. But here's the thing. Science doesn't deal in headlines. It deals in evidence, skepticism, and the slow, grinding elimination of every other possible explanation. And when it comes to Mars, we've been fooled before. So today, we're going to do something the headlines don't. We're going to sit with the data. We're going to understand exactly what Perseverance found, why it matters, why scientists are cautiously electrified, and what it will actually take to confirm or deny that we once shared this solar system with another form of life. To understand the discovery, you first need to understand why Perseverance went where it did. When NASA scientists were choosing a landing site, they weren't picking randomly. They were working backward from a question. If microbial life ever existed on Mars, where would it have left its mark? The answer pointed to one place above most others, the Jezero crater. Jezero sits in the northern hemisphere of Mars, and roughly 3.5 billion years ago, it was something extraordinary by Martian standards. It was a lake, not a frozen puddle or a temporary film of brine, but a genuine, sustained body of liquid water, fed by rivers, carved by deltas, lined with sediment. We can still see the ghosts of that ancient shoreline today, written into the landscape by orbital photography. The crater walls still bear the chemical signatures of clay minerals, materials that can only form through sustained contact with water. And where there's water, on Earth at least, there tends to be life. Perseverance, Percy to those who love it, touched down on the crater floor in February 2021 with a very specific mission. It wasn't there to dig trenches or set off explosions. It was there to read the rock record the way a geologist reads it on Earth, layer by layer, mineral by mineral, searching for chemistry that shouldn't exist unless something living put it there. Its instruments were extraordinarily precise. Its cameras could resolve features smaller than a millimeter, and it carried a core sample drill capable of extracting cylinders of Mars rock for storage and eventual transport back to Earth. Percy began its systematic survey of the crater, working outward toward the ancient river channels that had fed the lake billions of years before. In July of 2024, Percy was scouting the margins of an old Martian river channel called Cheyava Falls when its cameras flagged something worth a second look. The rock was part of what geologists call the Bright Angel Formation, a geological unit made up of ancient, water-altered sedimentary material. Two of Percy's most sophisticated instruments were brought to bear. PIXL, the planetary instrument for X-ray lithochemistry, uses finely focused X-rays to map the chemical composition of rock surfaces with extraordinary spatial precision. Sherlock, which stands for Scanning Habitable Environments with Raman and Luminescence for Organics and Chemicals, uses laser-based spectroscopy to detect the molecular fingerprints of organic compounds. Together, they form the most capable suite of life-hunting instruments ever sent to another planet. What they found in that rock, now named Cheyava Falls, was enough to flood inboxes at NASA's Jet Propulsion Laboratory within hours. The rock was laced with organic carbon compounds. It bore the texture and mineral composition of mudstone, the kind of fine-grained sediment that forms in still, slow water, the very environment where microbes on Earth flourish and get preserved. But the features that set pulses racing weren't the organics alone. They were a set of tiny, dark-rimmed spots embedded in the rock, ranging from 200 micrometers to roughly a millimeter across. The research team named them leopard spots and poppy seeds. Each leopard spot has a pale interior that is chemically similar to the surrounding rock, but its outer rim is markedly enriched with iron and phosphorus. That contrast, that sharp chemical boundary between center and edge, is not the kind of thing you typically get from random geological processes. On Earth, structures like that are almost always associated with one thing: microbial activity. More specifically, they appear to be evidence of localized iron reduction, a process where iron in one chemical state is converted to a lower oxidation state as a byproduct of metabolic activity. And buried within those spots, Percy's instruments found two minerals that made the astrobiologists sit up straight: vivianite and greigite. Vivianite is an iron phosphate mineral with a striking blue-green color when freshly exposed. On Earth, you find it in riverbanks, lake sediments, and near iron-rich ore deposits. And crucially, you find it wherever certain microorganisms are at work. Bacteria, such as Geobacter, are capable of a metabolic trick that most life cannot perform. They can breathe iron instead of oxygen. They take in iron in its oxidized form, iron 3 oxide, and expel iron in its reduced form, iron 2, as a metabolic waste product. This process is called chemosynthesis, and it doesn't require sunlight or a conventional atmosphere. These organisms can survive deep underground, in dark sediment, wherever iron and a carbon source are available. When the iron 2, they expel encounters phosphate and water in the surrounding environment, vivianite forms. That's the mineral signature those bacteria leave behind. Greigite tells a similar story, but implicates a different group of organisms. On Earth, sulfate-reducing microorganisms break down sulfate into sulfide. When that sulfide encounters iron in the rock, the two combine to form greigite, a magnetic iron sulfide mineral. The presence of both vivianite and greigite together in the same rock, co-located with organic carbon and within structures that have a distinct biological morphology, is not a combination that arises easily without life, at least not on Earth. The question the research team had to confront was whether that caveat matters on Mars. Good scientists are their own harshest critics. The Perseverance team knew that announcing potential alien biosignatures without rigorously attacking every alternative would be scientifically irresponsible. So they built the strongest possible case against themselves. The first alternative explanation was heat. Could volcanic or hydrothermal activity have produced the chemical reduction reactions responsible for these minerals without any microbial help? Sulfide, necessary to form greigite, can be generated abiotically when volcanic gases permeate groundwater. But there's a problem. That sulfide would have had to migrate from a high-temperature volcanic system into the much cooler, water-bearing sediment where Cheyava Falls formed. The surrounding geology shows no evidence of such a hydrothermal source nearby. There are no alteration minerals, no texture changes, no temperature signatures in the surrounding rock that point to the kind of heat needed. The geological neighborhood simply doesn't support it. The second alternative was purely chemical reduction, driven by organic matter reacting with sulfate. On paper, this can happen, but it requires temperatures between 150 and 200 degrees Celsius to proceed at any meaningful rate. The rocks surrounding the Bright Angel Formation preserve no trace of those temperatures. The minerals that would have been destroyed at such heat are still intact. The region was, by all geological evidence, cold. The third alternative was acidity. Under sufficiently acidic conditions, iron 3 and sulfate ions dissolve more readily into solution and can undergo reduction through purely inorganic chemistry. If early Martian water was far more acidic than current models suggest, some of these features might be explainable without life. And then Percy found the olivine. Nestled near the sample site was a small exposure of olivine, one of the most chemically vulnerable silicate minerals known to geology. Olivine is held together by electrostatic bonds between silicate ions and magnesium or iron cations. In acidic water, hydrogen ions flood in and displace those cations, dismantling the mineral rapidly. The more acidic the environment, the faster olivine breaks down. The fact that olivine was still present, intact and recognizable, was direct geological evidence that the water in this environment had never been particularly acidic. It was a small rock with a large implication. The acid hypothesis was dead. After months of analysis, the research team reached their conclusion. They could not demonstrate that the features in Cheyava Falls were produced by non-biological processes. Every alternative they tested either lacked supporting evidence or was actively contradicted by the surrounding geology. That is not a proof of life, but in the language of science, it is a very loud signal. Here is where intellectual honesty demands we slow down. Mars has promised us life before, and Mars has let us down before. And the history of those near misses should make us careful with our excitement. In 1976, NASA's Viking landers conducted the first direct experiments on Martian soil, specifically designed to detect microbial life. One of those experiments, the labeled release experiment, injected Martian soil with a nutrient solution tagged with radioactive carbon. The logic was straightforward. If microbes were present, they would metabolize the nutrients and exhale radioactive carbon dioxide. And to the team's astonishment, that is precisely what the instrument registered. The signal was positive. The lab was electric. Scientists who had spent their careers wondering whether Mars harbored life were suddenly confronted with data that said yes. But science is merciless. In 2008, the Phoenix lander revealed that Martian soil is saturated with perchlorate, a powerful oxidizing compound that, when heated and combined with organics, releases gases in a way that closely mimics biological activity. The Viking result wasn't life. It was chemistry pretending to be life. Then came 1996, and the meteorite ALH 84001, this piece of Martian rock, over 4 billion years old, and blasted free from the Martian surface by an ancient impact, ended up in Antarctica. When scientists examined it under an electron microscope, they saw shapes they could barely believe. Elongated structures, tubular and segmented, resembling fossilized bacteria. The discovery was significant enough that President Clinton addressed the nation from the south lawn of the White House. But by 2022, detailed geochemical modeling showed those shapes were most likely the product of a water-rock reaction called serpentinization, an abiotic process that can produce similar morphologies. Another signal extinguished. Mars has a habit of dangling possibility just within reach and then pulling it away. So when the Perseverance team says Cheyava Falls is the most compelling potential biosignature ever identified on Mars, we should listen carefully. But we should also remember that the most compelling is still not confirmed. There is one experiment that could resolve this question, and it's called the Mars Sample Return Mission. Percy has already done the first part. It drilled into Cheyava Falls and extracted a core sample of the mudstone, a cylindrical slice of rock about the width of a pencil and roughly 6 centimeters long. The team named this sample Sapphire Canyon. It is now sealed in a titanium tube, waiting. The Mars Sample Return Mission would retrieve it in 3 phases. A second spacecraft would land near Perseverance, collect the sample tubes, and launch them into Martian orbit using a small rocket. A third vehicle, already in orbit around Mars, would intercept that container and carry it all the way back to Earth. The entire operation would cost somewhere in the region of $11 billion, and the engineering challenges involved are genuinely formidable. The mission was announced as a joint collaboration between NASA and the European Space Agency in 2022. Since then, it has suffered repeated delays, budget overruns, and management restructurings. The target return date, once optimistically set for 2033, has drifted toward 2040, and more recently the project has been placed on indefinite hold. This despite the National Academy of Sciences, in its influential decadal survey, where leading researchers collectively determine the highest priorities for the next decade of space science, ranking the Mars sample return as the #1 mission priority, not once, but for 2 consecutive survey cycles. And that ranking was established before Cheyava Falls was ever discovered. If and when those samples arrive, the experiments scientists have in mind are extraordinarily precise. The first test would look for chirality. Amino acids, the building blocks of proteins, come in 2 mirror image forms, left-handed and right-handed. On Earth, life uses left-handed amino acids almost exclusively, while abiotic chemistry produces both forms in roughly equal proportions. If the Sapphire Canyon sample contains a strong chiral preference in one direction, that asymmetry is a powerful marker of biological origin. The second test would examine carbon isotope ratios. Carbon exists in different isotopic forms. Living organisms preferentially incorporate the lighter isotope, carbon-12, during metabolism, leading to a measurable enrichment relative to carbon-13 compared with non-biological materials. If the carbon in the Martian sample carries that biological fingerprint, it would be another brick in an increasingly solid wall. No single test will be sufficient on its own, but multiple independent lines of evidence, each pointing in the same direction, tested by different laboratories using different methods, would build a case that the scientific community could eventually accept. NASA has thought carefully about how to communicate confidence in life detection, precisely because the history of Mars research has shown how badly false certainty can damage public trust in science. In response, the agency proposed a framework called the Confidence of Life Detection Scale, a 7-level hierarchy of evidentiary strength. Level 1 represents an observation that is consistent with life, but could easily be explained otherwise. Level 7 requires multiple independent teams to have confirmed the same biological conclusion multiple times, using multiple methods, with no plausible alternative explanation remaining. The Cheyava Falls discovery, as it stands, sits somewhere in the lower middle of that scale. A genuine anomaly, chemically provocative, morphologically suggestive, and resistant to every abiotic explanation the research team could construct. But a genuine anomaly is not the same as a confirmed discovery. Returning the sample and running the isotope and chirality analyses would, if positive, push the confidence of life detection score significantly upward, potentially to level 6. Sending another mission to a different location on Mars and finding the same biosignatures independently would push it to 7. That is the definition of confirmed, scientifically speaking. It's a high bar. It should be. Let's do something the scientific papers cannot quite bring themselves to do. Let's reason through what it would mean if the sample comes back and every test is positive. The first and most immediate consequence is that Mars was hospitable to life far more recently than we imagined. Cheyava Falls is not ancient beyond reckoning. It comes from relatively young sedimentary material. That extends the window of Martian habitability considerably. But the deeper consequence is almost too large to process clearly. If life existed on Mars, then life has arisen in at least 2 places in our solar system, possibly independently, possibly through exchange via meteorite impacts, a process known as panspermia. If through panspermia, then we and any Martian microbes share an ancestor. If independently, then life is not a singular accident. It is a natural outcome of chemistry under the right conditions. That second possibility rewrites the Drake equation, the probabilistic framework used to estimate how many communicating civilizations might exist in the Milky Way. Within that equation is a variable called FL, the fraction of habitable planets that actually develop life. Current estimates for FL range from extremely optimistic to vanishingly small, depending on who you ask. If Mars turns out to have developed life independently from Earth, even microbial life, even ancient life, even extinct life, FL rises dramatically. 2 for 2 in our own solar system suggests that wherever liquid water persists, wherever energy gradients exist, wherever carbon chemistry can play, life may follow. And from there, the implications compound. If life is not rare, then the galaxy is not empty. The silence of the cosmos, which has haunted SETI researchers for decades, becomes stranger, not less. We would need new answers to old questions. Suppose the sample returns and every test comes back negative. Suppose Cheyava Falls turns out to be the most elaborate and misleading piece of abiotic chemistry in the solar system. Suppose Mars fooled us again. Even then, this story matters enormously. Mars is unlike Earth in one fundamental geological respect. It stopped. No plate tectonics, no continents sliding and subducting and grinding down. The Martian crust has sat essentially frozen for billions of years, preserving a record of planetary conditions from an era that Earth has entirely overwritten. The volcanic eruptions, the oceans, the impact events of the early solar system, all of that has been scraped clean on our planet by the restlessness of its own geology. But Mars holds on to it. Mars remembers. When we study Martian rock from 3.5 billion years ago, We are studying conditions that are analogous to early Earth, the world that existed just before life first appeared here. Even without confirmed biology, Cheyava Falls tells us something profound about how chemistry evolves in a water-rich, iron-rich, energy-rich environment. It tells us about the conditions under which life, if it were going to emerge, would find purchase. In that sense, Mars is not simply our neighbor. It is an archive. It is a record of everything our own world was before living things arrived and began to change it. For now, Sapphire Canyon sits in its tube on a cold, dusty plain, waiting for a ride home that may or may not come in our lifetimes. The science is not resolved. The politics of the mission are uncertain. The funding is contested. And the universe, as always, is in no particular hurry to give us answers. But consider what we now know that we didn't know before Percy drove up to Cheyava Falls on that July afternoon in 2024. We know that Mars, in the right place and the right rock, can produce a chemical fingerprint that looks like life. We know we cannot yet explain it without biology. We know the question is worth asking more loudly than we ever have before. The 0-1 infinity rule in astrobiology says there are only three options. Life exists nowhere. It exists in exactly one place, or it exists in so many places that counting becomes meaningless. We already know 0 is not the answer. And if Cheyava Falls is what some scientists think it is, then 2 becomes the next number. And after 2, the logic of the rule says the universe may be teeming. That thought, quiet, contained, still awaiting proof, is perhaps the most radical idea in the history of science. We are not alone. The cosmos, in its incomprehensible scale, made something else besides us. Something small, something ancient, something long extinct, but something. We don't know yet, but we are closer than we have ever been to finding out.